I'm Margaret Franzen from the Milwaukee School of Engineering Center for BioMolecular Modeling.
And I'm Cassidy Terrell from the University of Minnesota Rochester.
And we want to share with you a model that we developed under NSF funding for the CREST program. CREST stands for Connecting Researchers, Educators, and Students. It's a program that allows educators to work with the CBM to create models that are interactive and help in student learning. And this is a really cool model that Cassidy's worked on.
Yeah. So this is a model set that is designed to come after the enzymes content. So in my course, which I teach undergraduate biochemistry, I do enzymes content before lipids and membranes. And so I wanted to bring in a model that has both lipids and enzymes content so that I could refresh students on what they've previously worked with on the enzyme substrate, interactions content, and show them again that that content wasn't just a one-off. We continue to use that knowledge all the way through biochemistry.
Which is a really hard thing for students to accept. Right. I want to learn this chapter and forget it. And
right.
Yeah, okay.
Exactly. And so the cyclooxygenases, which have the abbreviation COX, COX, and there are actually a few different isoforms, but we're only going to talk about two. COX-1 and COX-2 are peripherally attached membrane proteins.
So this represents, the yellow represents the phospholipids in the membrane.
Yep.
This above the model is the inside, or you said this is in the endoplasmic reticulum.
Yes, it's the endoplasmic reticulum
and
membrane.
The enzyme only fits partly through the membrane.
Yes, so it's not a transmembrane protein that spans the whole membrane. It only sits peripheral to the membrane. So part of it is inside, part of it is outside. All the two cyclooxygenases, COX-1 and COX-2, are actually both dimers. They're both homodimers. So we've only shown one subunit because they're homodimers and the other one is identical. And so I'm going to kind of talk through how the features of this model, and there's a couple activities that go with it that I use in my classroom. But I wanted to bring back the enzyme substrate interaction content, tie it in with lipids, and also give students another example of structure-based drug design, or how knowing about the structure and function of an active site can lead to optimal design of drugs. So a little bit of the story of this. So the cyclooxygenases, like I said, sit peripheral in the membrane. This is their active site here, which contains both binding and catalytic residues. The substrate for this, for both enzymes, is the same. It is the fatty acid tail of a phospholipid, and that's arachidonic acid. It's got this unique shape here. So once this is cleaved from the phospholipid, it enters into the membrane, and the carboxylic acid here makes a favorable forked coplanar interaction with the arginine. And this arginine is the conserved binding residue, and that's going to be important later. The conserved catalytic residue is this tyrosine that is back here, and there's actually a heme group that sits a little bit in proximity to the tyrosine that helps facilitate a free radical mechanism that cyclizes this portion of the arachidonic acid and also oxidizes it. So there is a product molecule here, the prostaglandin, that also fits nicely in here. Again, it's still bound in. And this prostaglandin leaves the active site through the membrane and has a number of unique functions depending a little bit on its structure and oxidation around here. But primarily in the COX-1 enzyme, this molecule goes around the cell and just kind of helps with normal cell housekeeping functions. For COX-2, the COX-2 enzyme is induced only in times of pain and inflammation. So it is expressed, and its prostaglandin product signals pain and inflammation.
So this is showing the COX-1 enzyme,
yes,
which has a smaller binding pocket. And this is COX-2 that has this extra little pocket, and this would actually be a binding pocket in the active site. So these two enzymes are very similar. The COX-2 has a valine. The COX-1 has an isoleucine,
Exactly.
and the isoleucine fills or blocks this extra pocket.
Exactly.
Okay.
Yes, and that's something that researchers had noted as a difference between the two enzymes, because both of these enzymes are actually targeted by common pain medications that you could buy at Walgreens or Target or wherever you go. One of them is ibuprofen, and the other one is aspirin. So students are able to take, let's say, ibuprofen, and we can slide it into the active site. Again, it has this carboxylic acid that makes the favorable forked coplanar interaction, and it fits really nicely in the COX-1 active site. And if they switch it out, they notice that it also fits nicely in COX-2. So this will reduce pain and inflammation because it binds to COX-2, which means that if that is in and bound, the substrate can't fit. So, and this is an example of competitive inhibition because it is weakly bound and could leave again.
But because it binds to both the COX-1, the COX-2, and the COX-1,
yep.
it means that not only does aspirin turn off, ibuprofen, turn off the pain, it turns off the regular housekeeping as well.
Or just kind of reduces the amount. I have a hard time with offs and ons because it makes
That's
it seem
true.
like
So
nothing
it
is happening and everything is
slows
happening.
down the housekeeping and all the
Yes.
dust bunnies collect under your bed in the cell.
Right,
Yeah,
right,
okay.
yes, exactly. And the other common pain medication is that we have here is aspirin. And this one's really interesting too because it also comes in and fits equally well into both of the sites. But aspirin has this unique acetyl group here, and there is a serine that sits near the catalytic tyrosine that is actually able to attack the acetyl group and become acetylated itself. So the COX enzyme, COX-1 or COX-2, is covalently modified.
So it's a suicide inhibitor.
It's a suicide inhibitor. What's left over is salicylic acid, which is a competitive inhibitor. It's still inhibiting the entry of this substrate molecule, but this could leave. And this is still covalently modified. So even if the
this comes
substrate
in,
enters,
it can't do the normal reaction.
Exactly.
So no prostaglandins are made,
Exactly.
and then you have no pain, but you still have dust bunnies in your cell.
Exactly.
Okay.
And because aspirin is a suicide inhibitor with that covalent modification, it makes it a little bit more potent of a drug, which is responsible for some of its more negative side effects. So,
Can we design a better drug?
yes, hopefully. I mean, that was the idea.
Okay.
Okay, so the idea was, so now they know what the structure and function of COX-1 is, the researchers, and they also know the structure and function of COX-2. So in the rational drug design, the idea was, well, let's see if we can target just COX-2 and target only the pain and inflammation and not decrease the housekeeping functionalities of COX-1. So then there's this interesting discussion about, so if you know all of this, and the main difference between COX-1 and COX-2 is the shape of this active site, what would you do? How would you use your knowledge of the binding residues, the catalytic residues, the shape, the substrate, the product, the location of this in the cell to design a new molecule that selectively targets COX-2?
So you have to have something that interacts with this binding residue and
That's
forms a forked coplanar interaction.
Ideally.
You want to fill the part of the active site so that you block the access to the catalytic site.
Yep.
And since COX-2 but not COX-1 has this extra pocket, you want it to fill that pocket.
Good. Exactly. So that's pretty much exactly what researchers did. They kind of noticed that there's a bit of a V shape here, and knowing the interactions that can be made, a lot of the COX-2 specific inhibitors have a V shape. And they also, again, have to have a little bit of the hydrophobic quality because it has to enter in through a membrane. And so this can enter in through the membrane and sit in here. There's a couple different ways that this could fit. but students will keep rotating this around until you've made optimal contact with this. You could also, if we happen to have dots on the other side of this, we'll leave these halogens here, and this hydrophobic residue fits a little bit better when it's near this hydrophobic, this hydrophobic functional group fits a little bit better with this hydrophobic valine, and again, the halogens here are making interactions. I'm going to flip it over just so you can still see the color. And then once you have it in this formation, you could try and fit it into COX-1, and it won't.
It just won't fit in.
It just doesn't fit.
So then you get pain reduction,
Pain and inflammation reduction.
but
Yep.
you still get the dust bunnies out of your cell.
Right, exactly.
The housekeeping
Yes, hopefully you don't
responsibility.
get the stomach ulcers with the aspirin,
Right.
because you aren't inhibiting COX-1, and you are able to, reduce the pain and inflammation of, say if you cut yourself while you were slicing up vegetables.
Cool. Great. Thank you.